Yeah, I guess it works. Hi, I'm Fabi, and I'm going to talk about everything that's non-wavy about brain or body signals. So essentially, looking at how this broadband aperiodic 1 over f recorded using MEG, EEG, and ECG may be linked or not. So yeah, what's the 1 over f? What do I mean by that? So essentially, it has been shown, well, like it's often shown that electrophysiological activity is basically decreasing 1 over f. And when I'm talking about 1 over f broadband and aperiodic, what I mean is basically that the shape of the slope is shifting. All right, and this has been reported before by many very interesting studies showing that this was shifted in psychiatric disorders, different cognitive state, like in a working memory paradigm here, and also most prominently in aging. And this has been linked physiologically to excitation inhibition ratios, where more excitatory processes are having like a flatter spectra, and more inhibitory processes have a steeper spectrum. All right, but this 1 over f-like aperiodic activity is not only present in the brain. And interestingly, for instance, in the ECG, we can see similar effects that have been also reported for the EEG. So basically, these are like some 4 large data sets, and we see the same flattening with age that has been also reported in the EEG literature. So where does it lead us? Oh, this slide was different. Okay. So basically, what could this mean? So one hypothesis could be what we are measuring is really the same signal, but just measured at different locations, right? There's some volume conduction of the ECG, and we measure that at the head. Or it could be like a body-wide phenomena that cannot be reduced to volume conduction that's happening at the heart and also at the brain. Or both are just like 2 independent processes that just happen to be modulated similarly. So the first question that we then ask is, how strong is actually the ECG signal present in MEG and EEG recordings? So what we did is we took some resting state data where we have MEG, EEG, and ECG measured, and then we split the data basically in 3 conditions. One, removing only the ocular activity and keeping the ECG in. One where we remove both cardiac and ocular activity, and one where we only project back the ECG components. And then we try to decode the electric ECG time series doing that. And what we can see is that actually in both MEG and EEG, this is similarly strongly encoded in the signal, and but this is harder to remove kind of from the EEG than compared to the MEG signal. And most importantly, that happens at 0 time lags. So this speaks for some volume conduction here. So we then wanted to investigate how this would affect results, aging results for the 1 over f stuff. So we took like a large data set with some 600 subjects. We did the same splitting as before. But then I was faced with the question, okay, so but from where to where do we look at this aperiodic broadband activity? Because there seems to be like no really decided upon range in which we look into that. And when you look at the number of studies that are being conducted, I mean, there is some clustering here, right? But in general, there's like a wide range of frequency ranges being explored. You can not see it so well, but yeah. So what I did is I thought, okay, let's just analyze all frequency ranges. Basically, we split the periodic from the aperiodic stuff using ERASA, and then did slope fitting to 220 different frequency ranges, and then basically used that to predict age. So what you can see in the next slides are a lot of these plots. So basically, each like little parcel here is the result of a statistical test, and we did that for 20 different frequency ranges and also for every channel. So what do we see? We see a lot of not only flattening effects, but also steepening effects and also some null effects. When we look at the data when we didn't reject the ECG components, basically, where this clusters more in the low frequency range, the flattening, and the steepening is more all over the place and also in high frequency ranges. But there are also like a lot of null effects in the low frequency range. And when we remove these components, a lot of these like steepening effects disappear. These flattening effects in the low frequency range become more obvious, but we also get like a lot more of null effects in the end. And then when we look at the ECG components, we actually see the most effects. So we see most steepening effects, and we also see as a number of effects the most flattening effects. So when you summarize this differently, you can see that the ECG components, like that most of the observed effects are on these ECG components. But when you look at this, you can see that in some frequency ranges, like here, right, there are effects in all cases, right, in all different conditions. So we wanted to see whether these effects would be independent from each other. So what we did is we have here some ranges where we have unique effects for flattening and for steepening. There are no unique steepening effects in ECG-rejected data, and also unique effects for ECG components, and also these ranges where we have like shared effects. So what we did is we wanted to control for the influence of the other. So we used the ECG components as a covariate in a linear model, and to see whether these effects here are statistically independent. And in doing so, what we can see is like these three effects here in these three ranges all disappeared in both cases, whereas the steepening effects for the ECG components, they just stayed in the data. So to come back to our initial expectations, I think we find evidence for actually all three of these different scenarios, depending on where we look in frequency or location. And with that, I want to thank all the people that were involved in this work and all the participants, the data that I analyzed, and also especially to the people that get, I think, too little thanks, those that write code and make it available for us to use, because I think I wouldn't have come very far without them or without you. So thank you for that, and also thank you to the sponsors. And if you want more, I can also continue. Any questions?
Fabian, thanks for the presentation. So at the beginning, you kind of described what 1/f in EEG relates to. What is 1/f in ECG, though?
Brilliant question. I don't know.
No
I don't know.
idea.
No, I mean, I'm just like looking at the slope of the broadband spectrum. What I did is wait 1 second. I can show you some stuff. Yeah, wait. There it is. So, because I don't know, and I'm not that deep in the HRV field as many of you are, I was just naively using all the measures that I could find on Neural Toolkit, and then basically correlated these low frequency effects and high frequency effects of the ECG with all these measures, and there seems to be some relations there, but yeah. So it seems to be something that would be meaningfully related, I guess, to some heart rate variability. Thanks. Thank you.
Thanks a lot. I wanted to ask you about the ICA components that relate to the cardiac artifacts. How do you identify them? And in particular, the electromagnetic one and the pulse-related one? The one that shows the sharp RP component and the one that is slower in dynamics. And also the last question, do you find these components in all the participants or in just some of them? And what about MEG and EEG?
So this is a good question. So basically, how I identify these components, basically via correlation. So we measure ECG concurrently, and then we correlate the ECG time series with the basically with the independent components that we get from the data. And then we use different thresholds actually to get that. So basically, we don't have, like when we use a very high threshold actually to detect components where we can be like super sure, right, that it's only ECG, or mostly only ECG, I guess. We only find that in like 278 subjects, right? But we still get the same effects, like on that, independent of the sample, right? But so I'm using like a arguably low threshold. I mean, you can still see it in the time series very well. Yeah, does this answer your question? So, and the sharp peak that you saw before, basically what we did is we used a tool that we often use in auditory neuroscience called a temporal response function. It's similar to a cross correlation in a way, and this gives like this sharp peak basically with for the decoding.
Yeah, thank you, Fabian. Very nice. We were scrutinizing your paper on more than one occasion, of course. And each time I hear it, new thoughts come to my head. So now what I'm thinking is you are not really reporting a proportion of brain data which potentially could be accounted by ECG volume conducted stuff. So now I become more convinced that anything above 5 Hertz is unlikely to be ECG driven directly. Why? If this is ECG driven through volume conduction, then if you do coherence between ECG and brain signals, you simply must see very high coherence from 5 Hertz to whatever fitting range you had, let's say 30, 40 Hertz, okay? But it doesn't exist. So what it means is that it's unlikely to be coming directly from ECG. So what could be a common ground is that impedance of skin and skull is changing with age, and that similarly would affect it. But it would also explain why there is literally no coherence between ECG and brain starting from about 5 Hertz.
I agree with that part, if it were only EEG data. But we are measuring mainly MEG data, and there should be no effect of skin conductance, right, in the MEG, I would think. And probably also no effect of the skull, right?
But again, I don't think you will see coherence above 5 Hertz between ECG.
Okay. You mean you don't see
that
I don't see the
exactly.
coherence? Okay.
So, and the cool thing about coherence is even if you have completely aperiodic, like white noise signals, which are just a mixture, like linear mixture, you should have coherence at all frequencies. Yeah, but it doesn't exist.
That's a good point, yeah.
So what it means, I don't think starting from 5 Hertz, it's directly ECG driven. It's something else running in parallel for both of them, but it's not so simple.
Yeah, it's definitely not so simple. That's probably true.
Sometimes.
But I mean, that's what I also try to hint at. That's why I said like I get like evidence. I see some evidence for all these things, and then I guess we need more studies to really know what's going on in the end. So this is just like it's just the start.
Another thing to control. Yes, thank you.
Sorry, no, we can talk later if you want.
Yeah, of course. Oh, one more question, please.
So very interesting. I was wondering, well, every time I see this aperiodic component, many of us wonder what is the physiological meaning. And then in your case with aging, it's known that with aging, the body slows down, the brain, all the organs. So could it be that is what you're quantifying is just this less activity? That's it, not necessarily an aperiodic component, but it's just there is less activity in both brain and heart.
Yeah. I mean, it could be like, you mean like that there's in general like a drop in high frequency activity?
No, because you are looking for the slope.
Yeah.
But if you have another power spectrum, without looking at the slope, but with less activity, less power in your, you are likely to find us different in the slope as well. And that could be related to aging.
I can also show you something from my bonus slides, if I can, just very quickly. So basically, so we also have, as part of this working memory study that we included, where we just look at ECG. So basically, working memory effects of ECG, if you will. Basically, there's also like the same finding for the for where you have like a flatter slope and a delay period and a working memory paradigm compared to the baseline. And we took like an online data set where we basically have the baseline, and we have a task where subjects had to memorize sequences of numbers, and they were longer, meaning they were getting harder, right? And then the interesting thing here is that also we can also find the same flattening effect on the ECG data there. So this should not be like just aging, but I get your point. Like, I mean, for aging.
Yeah, I was just wondering if we can really disentangle this kind of obvious slowing down of activity with the aperiodic component. So it's kind of, it's a confound.
Yeah.
Yeah.
Sure.
Thank you.